Ha 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 ha. I am the copper ion. Me and my community of other metal ions have been working for the benefits of the cells since eternity. We are engaged in various biological functions like catalysis, metabolism, maintaining blood pressure, and neuronal signaling. Hello everyone, I am Kunika Gupta, a research scholar in the Department of Chemical Sciences at TIFR Mumbai. I am involved in the development of fluorescent probes for the detection of metal ions in the biological system, with my specific interest in copper ions. So my job is to spy on the copper ions in the biological system. Considering TIFR campus as the cell mimic, let's see what the copper ions are actually doing here. Copper ions perform catalytic actions when present in the bound form in the protein cores. They perform neuronal signaling in their labile form, and in their resting state, they are found bound to the glutathione. When proteins are mutated, copper ions find difficulty to get released from their site of action, or they are unable to enter their targeted organelles or enzymes, leading to their accumulation in one particular region. Further, conditions are severe when copper ions find their way to organelles or proteins where they are not even required. Also, we are graced with the supreme power of being invisible. Ha 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 ha! Disruption of copper homeostasis can lead to the accumulation of copper ions or displacement of copper ions from their desired location, causing some of the severe pathophysiological disorders like neurological diseases, Menkes disease or Wilson disease. And hence, it is very important to understand the localization and dynamics of the copper ions in the living biological systems. And for the detection of copper ions, we have utilized a strategy where we have developed a copper selective probe which has a selective scaffold which will bind to the copper ions, and upon binding of the copper ions to the scaffold, the fluorophore part of the probe will start glowing, telling us that indeed the copper selective probe is visualizing the copper ions inside the biological system. With this copper selective probe, now copper ions can be seen at their active sites, in their resting stages, and even when they are performing the neuronal signaling. Now from the fluorescence intensity, we can understand whether the copper ions are accumulated at one particular region or if their localization is different from where they are actually required. In my case, the probe that I have made for the selective detection of copper ions has a binding scaffold and a glowing moiety in its switched off state attached to it. Upon binding of the copper ions to the binding site, this glowing site will be released and will be in its switched on state. Now to demonstrate how this copper ion detection actually works, I'll show you a small demo inside the lab. Here we have taken the probe solution in both the cuvettes. Now if we bring in the copper ion solution and add it to the cuvette marked as probe, let us observe what will happen. So the copper ions are being taken and added to the cuvette. We do not observe any change here as the probe is not expected to show any color change in the visible region. Even after shaking the cuvette for a while, we do not see any difference between the two. But what if we start the UV light? Immediately you will see that the probe has started emitting in the green region where we have added the copper ions, thus proving the strategy for the detection of metal ions. And with this strategy, we were indeed able to develop a copper selective probe which can visualize the copper ions inside the living cells and the live zebrafish model. So this piece of work adds up to the early detection of the copper ions in the severe diseases caused by the accumulation or the misregulation of copper ions. I hope with this I was able to give you a glimpse of my PhD work. Thank you.